Okay, folks, I'm going to go through these cellular transport notes at full speed. So, normal talking speed. If you need to pause to slow down, then please do so. So, homeostasis. This is the need of an organism to maintain and regulate constant or stable internal conditions. Um, you can't answer this question, but how does your body regulate, aka maintain homeostasis? Some examples that often come up are things like sweating when it's hot or shivering when it's cold. But there are lots of different reasons and ways that your body maintains homeostasis. Temperature is only one of those. pH, so how acidic or how basic parts of your body are, and the concentration of other materials and nutrients. So, getting, say, sugars in and out of your cells, getting wastes like urine out of your body, need to be maintained within a relatively narrow margin. Much of homeostasis is maintained by the cell membrane, controlling movement of things in and out of the cell. So, a lot of the examples we think of, like, say, getting oxygen in and out of your body by the movement of your lungs, um, that's only part of the story because a lot of what then happens after that part that we notice, the movement of your chest to allow your lungs to expand, happens at the level of the cell membrane, allowing things in and out. The cell membrane, we talked about the cell membrane when we talked about cells and organelles a little bit. The cell membrane is said to be selectively permeable. And what that means is that certain substances can move freely across the cell membrane while others need to move through a gate. So this is not literally a gate in the cell membrane, so that's why it's in quotations, but it is a usually a protein that acts like a gate to control what goes in and out if it's something that isn't able to move freely on its own. Transport of materials across the membrane can be classified either as passive transport or active transport. So, passive transport requires no extra energy by the cell because molecules move from high concentration, being squished really close together, to low concentration, very spread out. That would mean that if they're moving from high to low concentration, they're moving down the concentration gradient, so high concentration to low. Active transport, on the other hand, requires extra energy, and that's in the form of ATP, something we'll talk about a lot after here shortly after we get through cellular transport. And that extra energy is being spent to bring materials into the cell or expel materials out of the cell. But in this case, when you're doing active transport, you're generally moving from low concentration to high concentration. So you're going against that concentration gradient. You're forcing that substance to pack more tightly together, which most substances, just like most humans, don't particularly want to do. They don't want to get packed more tightly together. So you're going to have to apply some energy to do that. Examples of transport. So, passive transport. One type of passive transport is diffusion, another type is osmosis, facilitated diffusion, and then the types of active transport we will talk about are molecular pumps, exocytosis, and endocytosis. Some words to know before we get started. Solute, this is what gets dissolved. Um, and I'm gonna use the example of making lemonade from powder. So if you're making lemonade out of powder, that would be the powder that you're gonna mix in with the lemonade flavor. A solvent, this is whatever does the dissolving. And basically always in biological systems, it's water. Same thing if you're making lemonade from powder, you're gonna use water as the solvent. And then the solution is once you've mixed those two together, the solute and the solvent, and now you have a uniform mixture, and that is your solution. And the example again in this case would be now you've put your lemonade powder in the water and stirred until you have a uniform mixture, and then you have lemonade. Concentration is the amount of solute dissolved in the solvent. So it's a measure of how much is in that amount of liquid. The symbol for um, so the symbol to abbreviate concentration are these kind of square brackets, and so we'll use those. You'll see those in upcoming slides. Let's talk first about passive transport. We'll go through our three different types, and then we'll move on to active transport. So diffusion first. And diffusion is the spreading out of molecules across a membrane until they are equally concentrated on both sides of the membrane. And again, all of this in biology is always talking about across a membrane, so either in or out of the cell. Molecules move down a concentration gradient because this is passive transport. There's no energy required. So you're going from high concentration, so remember that those brackets are the abbreviation for concentration, to an area of low concentration. So the molecules get to spread out. Example of some molecules that move through passive transport would be oxygen and carbon dioxide, because oxygen your body needs or your cells need oxygen. And then carbon dioxide is the waste product, so they want it to be able to freely get out. So here in this picture on the bottom left, you've got the dashed line representing a cell membrane. And then on one side, you've got lots of green solute dots. So like let's see, 2, 4, 6, 8 green solute dots. And then on the other side of the membrane, only 2 solute dots. So your left side is high concentration, your right side is low concentration. So think about which way the solute would go, and then the correct answer is that way. So from high to low concentration. And that would mean over time, as those molecules, those solute molecules moved from high to low, you would end up with an equal number of the solute on each side of the membrane. Now, facilitated diffusion is very similar to regular diffusion, but you need the involvement of a transport protein to act as a channel to help or facilitate the diffusion of molecules that normally couldn't pass through the cell membrane. Molecules are still moving down the concentration gradient from high concentration to low concentration. Some examples of molecules that move this way are glucose, which is sugar, and sodium or salt. And the reason glucose has to move through a protein channel and can't just go through regular diffusion is because of its size. It's just too big to go through the cell membrane on its own, so it just needs a tunnel to be able to get through. Otherwise, doesn't require energy and it's going from low concentration or high concentration to low concentration. Salt or sodium is small, but the problem is it actually has an electrical charge, believe it or not. And so because it has this electrical charge, it can't go without a channel either. So that's why each of those have to go this way. And so what you notice, this picture on the bottom left, pretty much the same as the first picture. You've got high concentration to the left of the membrane and low concentration to the right of the membrane. But now there's this additional orange tube in there, and that is the transport protein that's going to act like a channel or a tunnel. And so then over time, the solute molecules are still going to move from high concentration to low concentration until there are the same number of solute molecules on both sides of the membrane. All right, last type of passive transport, and we'll actually spend quite a bit of time looking at osmosis in a lab later this week. In osmosis, this is the diffusion of water across the cell membrane. So instead of the solute moving, the thing that is dissolved, now we're essentially talking about the solvent or the water being the thing that's going to move instead. And water molecules in osmosis are still going to move down their concentration gradient. So they're going to go from the area of high concentration, so the area with lots of water, to an area of low concentration, little water. And so what this looks like, now we have our membrane dividing an area with uh, low solute concentration on the left, so only three solute, red solute dots. But now, compared to the number of solutes, you actually have quite a bit of water. Compared to the right, now you've got lots of red solute dots, but that means there's less water on that right-hand side. So think about if the water is going to move from high concentration of water to an area of low concentration of water, which way would the water molecules go? And so they're going to go from high concentration of water to low concentration of water, even though that's the opposite of if you had to guess which way the red dots, the solutes, would go if they're able to, which sometimes in this case, they are solutes that can't go across the membrane. So the water has to go. The solutes would go the other way from the water. So then over time, what you end up with is it would actually raise the water level on this other side so that now, even though there's a greater volume of water, a greater amount of water, the concentration, so how spread apart the red dots, the solute is within the water, is the same on the left and the right hand side, even though the amount is different. Okay, so we need to talk about some vocabulary around osmosis before we go forward because the better you understand these couple of terms, the easier this unit will be to understand, and the lab we do will be easier to understand as well. So the first one is hypertonic solution. And so this is referring to whatever solution is outside of the cell. So, what solution does the cell find itself stuck in? And if the solution is hypertonic, it means the water concentration is below what is found in a cell's cytoplasm. And so, because the water will tend to move out of the cell because the cell is hypertonic compared to the solution. So think about hypertonic, like hyperactive. When you're hyperactive, you have too much energy. That means the cell has too much water compared to what's outside of it because the water concentration outside is below the cell. Then the water is going to tend to move out of the cell down its concentration gradients from high water, hyper water inside the cell, to lower water outside. And what happens in that kind of a solution when you stick a cell in that solution is it's going to tend to shrivel up and shrink. So think about hyperactive, hypertonic means lots of water in the cell. Hypotonic solutions. This is where the water concentration outside of the cell is above what's inside. So there's less water in the cell, more outside. And so the water will tend to move into the cell down its concentration gradient because, again, there's more outside, less inside. And so the cell is going to tend to swell up like a balloon. Isotonic solutions, this is where there's an identical water concentration on the inside versus the outside. And so then the cell is going to stay the same. So let's look at this kind of slightly more graphically. So hypertonic, remember the cell has more water, so it's hyper on the inside. And so water's going to tend to move out. And then what happens is you get a cell that shrivels up, like, say, a raisin or something like that. Hypotonic This is where the cell has less water inside, and so there's more water outside. So the water is going to tend to move in to the cell. And in this case, the cell is going to swell, and actually, in some cases, might even swell until it bursts. Isotonic is where there's the same amount of, or the same concentration of water inside versus outside. And so water will still flow in and out of the cell, but in equal amounts. So the amount flowing out and the amount flowing in will balance each other out, and everything will be the same. The cell will stay the same. Okay, moving on to active transport. Remember, not all substances can move so freely into or out of a cell. Some molecules have to be pushed uphill against a concentration gradient, so they have to be packed in more closely together. And so this means substances are moving from an area of low concentration to an area of high concentration. On the other hand, some substances are just too big to move through a protein channel. So even if they might be going with their concentration gradient or down their concentration gradient, it doesn't matter. They're just plain too big. And so they have to be enveloped by the cell or excreted by the cell in a different way. They're not going to fit out of that channel. And this is where we get into the endocytosis or exocytosis that we'll talk about in a minute. First, though, we're going to talk about molecular pumps. So this is where we're going against the concentration gradient. So the molecules aren't necessarily giant, but you're going the wrong direction. A cell has to use energy in order to pump these molecules across the membrane through a protein channel. So remember, you have protein channels in passive transport as long as the cells were going down the concentration gradient. But now, if you want the cells to go against it through a protein channel, you've got to use energy. And why the cell would even do this is it allows cells to concentrate key or important molecules within the cell or remove waste quickly because some kinds of waste is really toxic to the cell and it doesn't want to wait until it can go down the concentration gradient. It may want to push it out. Even if it wastes the cell's energy, it may be too toxic and it's worth it to push it out. Some examples, things that cells often want to concentrate, things like calcium, potassium, chlorine, sodium, because these are ions, they're charged particles, and they are really key molecules within a cell. So even though they could move through a protein channel down the concentration gradient, the cell may not want to wait. It may want to concentrate them on one side or the other. And so in this case, same picture, you've got on the left side, high concentration, on the right side of the membrane, a lower concentration of solutes, but this time the cell is going to put in energy to send those solutes against the concentration gradient so that now the left-hand side is even more concentrated and the right-hand side is even less concentrated. Now, I mentioned that sometimes the reason a cell has to move things using energy has nothing to do with the concentration gradient either way. Sometimes a cell is just plain too big to go. And this is where a cell uses energy to import large amounts of material into the cell using a vesicle. So remember talking about vesicles when we talked about cells and organelles. Now here's part of their job. Um, so this can be large amounts or actually physically very large substances into the cell. So endocytosis is going in. It's almost like endocytosis, but not quite, unfortunately. And a good example of this is a white blood cell engulfs or eats a bacteria to fight infection. So let's say you fall down, you skin your knee on a really dirty surface playing basketball or something like that. And now you've got bacteria from the gym floor or the outdoor basketball court that's in your body. Your body's going to send these white blood cells to almost almost really like a Pac-Man in a way, eat those bacteria so that your body can then heal your knee and you don't get an infection from that. And so here, the arrow showing the substance going in, it's almost like sort of the cell membrane dents in, and then bloop, it folds around the particles it wants to take in, and then it takes them into a vesicle. And then, depending on what it is, maybe the vesicle, if it's a product the cell wants, it might take it to one of the organelles to use. Or if it's a bacteria, the vesicle might fuse with, say, a lysosome so that the bacteria can get digested. So, what happens after it comes in through endocytosis kind of depends on what that stuff is. The opposite of this process is exocytosis. So, endo is like endocytosis, and exocytosis is exit cytosis. So, leaving the cell, exiting the cell. And this is where a cell uses energy to export large amounts of material out of a cell using a vesicle. So again, the vesicles, this time, whatever is in that vesicle has been made maybe in the endoplasmic reticulum, maybe in the Golgi apparatus, has done some final processing of that substance, sent it out into a vesicle. That vesicle then goes along, gets closer to the cell membrane until finally it fuses with the cell membrane and whoop, the contents go out of the cell. And an example of this would be nerve cells release neurotransmitters. And you'd want to do that so that if you're a cell that communicates with the brain, you can send those neurotransmitters and they can move up to the brain rather than keeping your message or your signal secret inside your cell. And then that's no good. The brain doesn't know. So that's a reason you want to make something inside your cell, maybe, and then send it out. And again, that's just one example. So now it's practice time. So go to the table at the bottom of the second page of the notes. Check out the table there. Do your best to fill it in from your notes, and then we can check it together later.